Thank you, Dee Dee, and thank you to the organizers, and thanks everyone for coming here. It's really so an honor and a challenge to talk after Dr. Monto, who has just sort of a mastery of this field that is so impressive, and actually it really is a great step to what I'm going to say. And I'll probably go kind of quickly through some of the background which Dr. Monto has presented. I'll talk a little bit about both seasonal and pandemic vaccines, and predominantly focusing on the sorts of incremental improvements that I think at least most of us in industry, as well as those in academics, agree are needed and certainly possible. And I will end up with some of my own views on universal flu vaccines, which again I think you'll find pretty consonant with Dr. Monto's. Let's see if I got this right. Okay, not sure where do I point this thing.
I think it's just down there.
Just down here. Okay, great. All right. So first, a disclaimer. I guess I am a businessman as well as a scientist. So here's the disclaimer. I'm a corporate stooge, and make no bones about it. We make and market influenza vaccines as well as do science on flu vaccines. The need for improvement comes both from the seasonal side and certainly was highlighted, as Dr. Monto mentioned, the 2009 pandemic, and that resulted in a report from PCAST, the scientific advisors to the President, about things that could be done to improve our pandemic preparedness. And in fact, we've really taken that as a guide and tried to do many of those things. So I'll talk a little bit at the end about some of the more radical things with RNA-based flu vaccines, which are preclinical at this point. I'll talk about our efforts on pre-pandemic vaccines to implement cell culture in U.S. vaccine manufacturing, use of adjuvants, and particularly use of synthetic biology to improve not only the speed, but we also think the strain match of vaccines that will be out there soon. Right. And even to this day, although in 1943 there were influenza vaccines out there, what is striking is 60, more than 60, 70? Good Lord, 70 years later, influenza remains the most variable pathogen against which we have any effective vaccine. The non-variable pathogens, for the most part, except for some special cases like RSV, for the most part, we actually have effective vaccines, and for some of them, we've had them for a very, very long time. You could add rabies to this list and get back into the 19th century. Whereas when you get to pneumococcus and meningococcus, these are pathogens that sort of change at a moderate rate, so you have to change the vaccine every now and again. Every 10 years or so, you have some sort of an update, but it's pretty not that hard to keep pace with them. And at this end, you have HIV, and you could add HCV, hepatitis C virus, to this as well. And when you get to the kind of rate you see there, we simply are today unable to make effective vaccines. So although the first effective flu vaccine, came out about 1943, all this time later, it's still the biggest variability challenge against which we can actually have a commercially and public health useful vaccine. It all comes down in some ways, it doesn't all come down, and certainly the universal flu vaccine efforts, we would dispute that it all comes down to this, but our current vaccines largely come down to the properties of hemagglutinin, which remains the primary target, although certainly neuraminidase and conserved interior antigens are also secondary potential targets for vaccines. And the core issue is the most potent immunity is directed against this receptor binding head up here. And that's the part that changes a lot, not surprisingly. It's where the most powerful immunity is directed against. It's under the greatest immune selection pressure, and it changes all the time. And so we're chasing the changes in structure in the HA head. And that results in the full complexity of the current influenza vaccine system. We have to change a lot, and when you're a manufacturer, there is no other mass-marketed biological product. That changes every year. There are emerging biological products, for example, when you actually take people's own T cells out of their bodies and program them and reinfuse them in therapeutic contexts, but those are boutique products. And the standard pharmaceutical manufacturing is you go through a development program, you make your vaccine, and then every batch from there on in is made exactly the same way, and you have no variability whatsoever. Flu is different. Flu changes almost every year. And that poses huge problems for manufacturing, huge challenges, and it links together surveillance, manufacturing, and a giant public-private consortium to get this task accomplished every year in a way that I don't think is parallel for really anything else out there right now. And it's actually, in addition to the scientific interest in all this, there's a systems interest. Is how do you get a system like this to work and to get it to work better? In addition to seasonal influenza, of course, we've had the pandemics that have come with successive introduction of new types, or sometimes reintroduction of types. And what we've noticed up here is all these activity up here, much of which probably reflects increased surveillance. Some of it could reflect changes in human behavior, more interaction of human animals, and more human transportation around the world. But largely, I think we're much more aware of the constant probing of the human population by zoonotic influenza viruses, and particularly because of the potential of highly pathogenic strains, particularly H5N1, but also some H7N7 and other strains. There's increased attention to the desire to actually do something in preparation for the possibility that these could start to spread efficiently from person to person. What is so striking, and what really struck me, I was an academic before I came to industry, and when I first sort of wandered into the influenza vaccine business, it was like you're stepping back to those old journals, 1950s, 1940s journals, because I'd go to meetings, and they'd say, well, should we be using the guinea pig red blood cells, the turkey red blood cells this year? Should we be ether extracting? And it was all about HI assays, a release test that is essentially an Aucherlani test, which most people who are students in science may never even have heard of, because we have ELISA's, a much better test now. But that, in fact, is the technology that continues to dominate influenza vaccines. The real milestones were in the 30s and 40s when you figured out you could grow flu viruses in eggs. A big advance in 1964 when you realized you could use not just the whole influenza virus, but you could use part of it, which did not improve vaccine efficacy at all, but improved tolerability. And then the 1960s, this sort of ferment of innovation in flu vaccines. And at that time, top scientists worked on influenza vaccines. They developed not only splitting, but in the Soviet Union, they developed cold-adaptive live attenuated vaccines, the predecessors of, not the direct predecessors, but the conceptual predecessors of FluMist. The notion that you could purify virus by continuous flow centrifugation, what remains today a remarkable device that improves viral purity and vaccine purity, and still the dominant way that flu vaccines are made. And then the notion that you could genetically manipulate flu viruses to make them better substrates for making vaccines, not by genetic engineering, because this was 1969, but by mating flu viruses. So you could combine the characteristics of different viruses to get one that both has the antigenic characteristics you want and also the growth characteristics that you want. And then things got really quiet. In 1977, after the Fort Dix swine flu, there was a little change in one of the assays that we use. In other words, we moved from quantifying the amount of hemagglutinin in a vaccine from hemagglutination to SRID, which is this modified Aucherlani test. And for those who don't know what an Aucherlani test is, it's you have an agarose gel and you punch two holes in it, and in one well you put your antigen, and the other you put your antibody, and they diffuse towards each other and make a precipitin line. The test is a little different than that. You actually basically cook the antibody into the gel, but it's essentially the same concept. And that remains to this day the way that seasonal flu vaccines are quantified. And there are huge problems with it. There are questions of whether it performs as we believe it does, and you have to immunize a sheep every year to release your vaccine. So literally, many, many years, we have vaccine bulks sitting in giant cold rooms waiting for the sheep to seroconvert. I mean, it's not a very 21st century kind of thing to do, but it is what we do. And then innovation started a little bit in Europe. in 1997 when adjuvanted flu vaccines started there, although they're still not very dominant in Europe at all. There are still no adjuvanted flu vaccines in the United States to this day. And cell culture manufacturing started in Europe in 2007 and only came here much later. So there was really essentially from the 1960s until post 9/11, there was a 20, 30-year hiatus in flu vaccine innovation, in part driven by low prices, by indifference and the policies that no vaccine would be favored over any other, really robbing the incentive to innovate. And what changed things was not really a change in the business environment. It was a change in attitudes in the United States about security and biodefense. After 9/11 and the anthrax attacks, BARDA got involved, as Dr. Monto mentioned, and started funding innovation in flu vaccines. And that created incentive for many small biotechs to innovate, and a couple of manufacturers. The big manufacturers were a bit less interested, although Novartis, as it turns out, was. Maybe in part because its legacy, it was a biotech. It was Chiron and then became part of Big Pharma and maybe maintained a little bit of the biotech attitude about some things. And BARDA funded a wave of innovation that we see now that includes some of what I'll describe today, but also insect cell expressed recombinant proteins, increasingly attempts at universal flu vaccines, nanoparticle-based vaccines, etc. And so it really is an episode where the government actually had an intervention that really has had a real effect. And actually one of the challenges we face now is as that funding starts to drop, can the business environment sustain that innovation? Or once government sponsorship, if it does, we'll have to see what Ebola does to all of this, but if government funding drops as it has in the past few years, will the innovation be sustained, or is everyone just going to go back to making the cheapest egg-based product that they can? So just a little bit about some of the other basic technologies. We start off with whole virus and activated vaccines. There was a discovery that either through detergent or in some case organic solvent extraction, you could take out the surface antigens and get rid of most of the core, including the RNA, which actually made the vaccines more immunogenic, but also quite reactogenic often. And then in the 70s, started to get more purified vaccines. And then today is this ferment that I mentioned to you, including tetravalent, which is quite a conservative change, but also virus-like particles, recombinant vaccines, and the attempts at universal vaccines. So why not just keep making the cheapest flu vaccine you can make in eggs using this old technology? The infrastructure is there. We've been doing it for many years. What's wrong with it? Well, think about almost a billion fertilized eggs a year. This is a huge logistical issue. It is particularly a concern in a potential pandemic because although now the U.S. government actually pays for big egg production to make sure we always have a supply of eggs, that supply is vulnerable. You can't scale up fertilized chicken eggs that fast because you got to get chickens to do a bunch of stuff. And not all strains grow that well on eggs. And I'll talk about that in a little bit more detail later. And there have been failures of production of vaccines that are relatively well matched because you couldn't get a well-matched strain to grow on eggs in time. There's a huge waste disposal requirement. If you go, David Markowitz has spent time on our Siena site, and you know the smell on that site. I mean, we use about 100 million eggs a year at Novartis, and 100 million rotten eggs, that kind of smells. Although actually we do recycle. We make fertilizer out of the used eggs after you autoclave them. And one of the key things I'll talk more about is that all the production strains must undergo egg adaptation, and that has consequences for their antigenicity and strain match. And I don't think that even today we quite know the magnitude of that effect on vaccine efficacy. So lest you think that this notion that the egg supply is vulnerable in a pandemic is a complete fantasy, because certainly the biodefense world is full of paranoid fantasies. There is some basis for it. In 2012, I believe, there was a H7N3 outbreak in Mexico that led to a large-scale culling of flocks in Mexico and egg shortage, actually, in the country. And it actually did cause very limited human disease. There's conjunctivitis in a few cases. It was not severe disease. But it was an episode where there was a strain that was highly pathogenic for birds, resulted in an egg shortage, and caused some level of human disease. Now, had you had a highly pathogenic strain causing loss of chicken flocks and eggs and causing not mild human disease in a few cases, but severe human disease in many cases, you would have had the kind of scenario that biodefense planners and BARDA and those people really worry about. So there is some basis for thinking that this sort of preparedness is for a serious person, and getting eggs out of the system for that reason matters. One of the key points is that the viruses that are collected to make influenza vaccines come out of somebody's nose. And when they come out of someone's nose, they are adapted to grow in people. And in people, they bind predominantly alpha-2,6 sialic acid as their receptor. The vaccines are made in the allantoic cavity of chicken eggs, where the dominant receptor is alpha-2,3 sialicide. But why does that matter? Well, I showed you that picture of hemagglutinin where the head, the variable head, is the part of the molecule against which the most potent immunity is raised. And one of the key mechanisms of immunity is blocking receptor binding. Not the only one, but a key one. When you change your receptor specificity, you're changing the sequence around that receptor binding site. And therefore, in many cases, you can probably say in most cases, there is some degree of antigenic change that is associated with moving from human adaptation to egg adaptation. The degree of that antigenic change varies depending on what the change is and how disruptive that mutation is, particularly, for example, if it introduces a glycan. And therefore, essentially all vaccines in the US, and I'll get to why even those that don't need to be this way, it is that way, are mismatched to some degree or another on the basis of egg adaptation, even when it's technologically possible not to have them to be mismatched. And this is getting to the other point. So every year, you have some degree of mismatch on the basis of egg adaptation. In some years, you actually don't get a well-matched strain. 2004, the Fujian strain is the classic example of that. But the problem has not gone away by any means. This is a rather extraordinary slide showing how biological change in the virus has affected things. So actually, when WHO, National Influenza Centers, and I'll describe the system a little bit more in a bit, do their surveillance, they generally actually try to isolate viruses on mammalian cells, typically these dog kidney cells, MDCK cells, and the virus grows pretty well on them. But because the system is designed to work on eggs, other WHO laboratories, the collaborating centers, then if they want to actually use that virus to distribute to manufacturers, will go back to the original respiratory specimen and try to re-isolate the virus on eggs. When they do that, particularly for the H3N2 strains, the success rate is pathetic at times. This is what it used to be. Most strains that were being isolated in MDCK cells could be isolated in eggs, and it plummeted. For this period, around 2008 to 2012, I'm told it's getting a little bit better now. And this was a biological change in the virus. It got less good at growing, in the strains that were dominantly circulating in humans for H3N2 got less good at replicating in eggs. And so you had terrible isolation rates, severely restricting the available viruses to use in vaccines. Now, I'm going to go through just a little bit of data. And this shows, so if you try to go back and figure out how severe is the problem of egg-based strain mismatch, it is extremely difficult to do. Because if you go back in the literature more than about 2012, they didn't say. Now you see on this side, I'm not going to go through every number here, don't worry, but what I do want to point out is that there are two viruses here named A Victoria 351-2011. They have the same name. The difference is a little column that they started adding to these just in the past few years, showing egg passages versus cell passages. These viruses are actually quite different. They have the same name, but they're different. And here, if you look at this is a ferret antisera looking at HI tests of how similar the antigenicity is, and you can just see that 40 is a very different number than 1,280. Those are the antigenic differences by that particular comparison, depending whether you passage that virus in eggs or in MDCK cells. Now, if you look across, not all of them are as big, but actually, if you actually look from the time that you could get the information, you see consistently the viruses that are made available for vaccines are generally quite significantly mismatched to the actual strains on the basis of egg adaptation. I looked through the past 4 or 5 years where I could get the data, and 2 out of the 3 strains for the trivalent vaccine have been quite mismatched. H1N1 has been the exception. And what's so striking is this is in years where the CDC will make statements saying, we have a good strain match again this year. What do they mean by that? Many people might think that what they mean is the vaccine matches the strains circulating in humans. That's not what it means. It means that the World Health Organization recommended a strain and the manufactured a strain that matched what the World Health Organization recommended. It doesn't mean that what was recommended actually matches what's circulating. And actually trying to peel off the layers to get to the bottom of this is not very easy. The only reason I know this, and I'll get to this later, is we started synthesizing our own viruses. And we were set tests by BARDA and by CDC saying, your job is to show that you can produce better matched strains than the World Health Organization. And then we started doing it. And we realized we started going directly to what was out there circulating and matching it. And we found we no longer matched the strains we were being provided by the World Health Organization. We looked why. And we realized that the strains we were getting from the World Health Organization were all egg adapted. We were making strains that actually matched what was circulating. And they no longer matched what was recommended for the vaccines. It wasn't until we actually went to the lab and tried to make well-matching viruses ourselves that we realized that what was being recommended wasn't well-matching. So just reading the literature, you don't get this. You actually have to do the experiments to figure some of this out. Dr. Monto has already gone through this quite a bit. I'll simply say that there is a gap between vaccine efficacy. And you can look at many different studies, and you clearly have to look at them very carefully to piece apart how big that gap is. But everyone agrees that there's a significant gap. Everyone also agrees that a better strain match is better than a worse strain match. What I don't think any of us can say to this day is how much antigenic difference translates into how much difference in efficacy. So obviously we want to improve strain match. Well, we can't, and although we have a belief that that will improve vaccine efficacy, at this time I don't think we can say exactly how much of that gap between 50% and the 95% we wish we had, how much of that gap can be closed by improving strain match. I think we're going to have to find that out empirically. But to some degree, the test has never been run. It is not, really well-matched flu vaccines have not been available because everything is adapted to eggs. So this is our Liverpool site. We don't actually have the chickens on site, but we do have the eggs on site. And you can sort of see what a process this is. You actually have essentially 100 million little bioreactors that you're inoculating to make your flu vaccine. I mean, it is a triumph of industrialization that, you can do something 100 million times in serial to do it. But it's not a very efficient way to do things. So I'm going to go and talk a little bit about the system and then come back from there. Because of the strain change and the change in the virus, a surveillance system was set up shortly after the Second World War. After those 1943 introduction, it was just a few years later when vaccine effectiveness dropped. Dr. Monto showed the data. And when the drifted variant viruses were included in subsequent vaccines, effectiveness was restored. And in 1948, the World Influenza Laboratory was established in London to search for the dominant viruses. Today, this is expanded into a huge international system. In fact, this slide underestimates it. There are 5, and there may be 6 WHO collaborating centers now, with introductions in China as well. And there are now, I believe, over 140 national influenza centers. There are also the essential regulatory laboratories. What is this giant system? The national influenza centers are scattered around the world. They swab people's noses and throats. And they isolate viruses, and they try to see, using just hemagglutination inhibition tests, are there viruses that don't react with ferret antisera raised against the current vaccine strains and other recent reference strains. Why ferrets? Because ferrets get the flu. So you don't need to prepare an antigen preparation and immunize like you would in a mouse or a goat or anything else. That you can simply take a preparation of the virus, put it in the nose of the ferret will get sick, and a week or two later you got an antiserum. It's a very efficient system. So they take the red blood cells that may come from turkeys or chickens or guinea pigs, they take the ferret antisera, they mix them together, and they see, are there strains now circulating that no longer react well with the ferret antisera? And that says there's a new strain out here we have to pay attention to because it's antigenically different. That's all done with MDCK cells. Then they take this thing and they send it to the WHO collaborating centers. Those are more sophisticated laboratories that have the capability of doing sequencing, more sophisticated antigenic testing, generating their own ferret antisera, etc. And they will do antigenic testing. Now, of course, now you're shipping human respiratory secretions around the world to do this. If they find something interesting, either that center or another set of laboratories, the reassortant laboratories, which include some WHO collaborating centers, will then take that virus and in eggs attempt to mate it with another virus, particularly for the A strains, not so much for the Bs, to see if they can improve its growth characteristics. Then a group will get together in Geneva twice a year, once for Northern Hemisphere and once for Southern Hemisphere, and behind closed doors, I'm sure they smoke cigars, they will come up. Oh, they're women. Well, they can smoke cigars too. And they come up with a strain recommendation, and there are data released after that. Then some of these viruses go to manufacturers. The manufacturers make antigen, and some of this antigen is then sent to another set of WHO laboratories, the essential regulatory laboratories. And they're the ones that immunize the sheep. And after they've immunized the sheep, and some of these sheep get immunized again and again and again and bled again and again in a season, the sheep antisera go back to the manufacturers and they do this Ancelani test on the vaccine batch they've made to say how much hemagglutinin is there. And there's another very mysterious process by which, it's just not by comparing SDS page gels, it sort of gives the lie to much of the system. They decide how to calibrate these sheep antiserum and an antigen standard. So you can see from this system, it is wildly inefficient. It involves chickens and turkeys and ferrets and sheep. It involves sophisticated instruments like rulers and magnifying glasses. It is a system that was developed shortly after World War II in the suburbs of London, and it involves technology and reagents that you could obtain in the suburbs of London after World War II. And so this is a system ripe for improvement. So this is just, this is a picture. This is the world, and that's where the National Influenza Centers are. And this is just a little timeline that describes a little bit of what I've described back and forth. Lots and lots of shipment of stuff back and forth around the world. Not a way to do things quickly. So in 2009, this happened. This was an NSA photograph that actually shows the origin of the 2009 pandemic. And it shook up the system in a way for exactly the reason that Dr. Monto really outlined at the beginning of his talk. And that is, we have this system, and it was very nice when Dee Dee said, the fastest vaccine response in history, and yes, we're very proud of it. However, it is also true that it wasn't nearly fast enough. As Dr. Monto said, vaccine was not available in any substantial quantities until after the second pandemic wave. In other words, things started, kids went to spring break in Mexico, they exchanged fluids, they came back, you had your first little wave in places like New York. Things got quiet over the summer when all the kids went home from school. They came back in the fall, you had another wave. It peaked. You had spread all over the world, largely following airline routes, which was really very impressive. So then you got your second wave peak, and then the vaccine came out. Now, my corporate minders had me take the scale off of this axis that says how many doses were made. So it almost looks like, gee, that vaccine came out and it looked like it ended that epidemic. But no, that's not true. There were nowhere near enough doses out there to have had any significant impact on the pandemic. Now, I do think that vaccine did do good. I'm definitely not going to be an extreme vaccine denigrator. in 2010, not everyone got infected in 2009. And so I think that vaccine did do a lot of good, primarily in preventing infections in 2010. But in 2009, it really came out too late to have much of an impact. So we have worked with BARDA for some time, and I'll talk a little bit about the flu cell culture facility that we built with them and flu cell culture manufacturing. I'm going to start actually by talking about our initial response to the 2009 outbreak. The standard first step in making a flu vaccine is that you wait by your mailbox for the WHO to send you a strain. And you can wait a very long time. When the 2009 outbreak occurred, there was a new kid on the block, and that new kid on the block was BARDA. And BARDA was not playing by the rules that the other agencies that have been involved in flu vaccines for a very long time were playing by. They said, look, Novartis, we've given you a ton of money for pandemic preparedness. There's now a strain that was in Mexico and is now showing up in California. We expect you to do something about it and earn all that money that we gave you. So it was a very direct thing. On, I think it was October 24th of 2009, my boss called me. He had just met with Robin Robinson, who was head of BARDA, and Robin had said to him, do something, basically. And he called me and he said, do something. And he said, by the way, Robin says that somewhere on the web there's a sequence for this new virus. And so, you do a Google search, and try to find this thing. And in fact, it is more organized than that. There's a system called GISED that actually distributes influenza virus sequences. And when you go on, you click on a little something that you agree to agreement. So in the middle of the night, I was waking up lawyers saying, can I click on this thing? And the lawyers woke up and they said, yes, you can click on it. And so we clicked on it, we got the sequence, and we decided we were going to try to make our own vaccine virus while we were waiting for the virus to come from the World Health Organization. We had not had reverse genetics instituted beyond a graduate student who worked on basically mouse immunology studies. We had not cloned the standard backbone, genetic backbone for vaccines at that time. We certainly didn't have any synthetic capability. So we tried to synthesize a gene. We actually, on a Sunday, got a German factory to staff their gene synthesis factory and start work. We were never able to make a virus for that gene. The CDC, in the second week that this thing had become a sort of a public item in the US, sent us a viral RNA. We cloned it, and while we were at it, we subcloned the PR8 backbone into appropriate genetic vectors. And by about 14 days, or I think it was 14 or 17 days into the outbreak, we had the first potential vaccine virus anywhere in the world that we had made by reverse genetics and cloning standard laboratory cells. Now, we couldn't use that. It had been made in a research laboratory. It had not been made in cells that are qualified for vaccine manufacture. So it would not be suitable in terms of its quality standards to be used in the production of a human product or product to be injected into humans. But we didn't stop because we didn't have anything yet from the World Health Organization. So we thought, well, we're going to keep going. So we went to our pals at the University of Marburg in Germany, where they have a BSL-4 lab. And we didn't need a BSL-4 lab. We needed a highly quality control lab. And the kind of protocols that are used to run BSL-4 labs are a lot like manufacturing under quality control. Rigid protocols, careful environmental monitoring, protocols on how you decontaminate thoroughly, etc. We adapted the reverse genetic system to our work in our manufacturing cell line. And we actually came up with a reverse genetics virus that we believe would have met GMP standards. 2 days before, we actually got a seed from the World Health Organization system. And of course, we never used this virus because the regulatory risk of something made by a new process if something made by the old process was actually available was just too great. And in a real situation, like a pandemic, you can't afford that kind of risk. It really has to work. So we abandoned it at that point. But it taught us a lesson. Oh, by the way, if I may, so that is when we got the seed virus. When did we get the calibrated sheep antiserum? Way out here. We actually started a clinical trial before then. We couldn't wait for the sheep antiserum to arrive to start our trial, so we made up our own assay, and we gambled that when the real assay came out from WHO, our assay would match it more or less, because we need an assay that didn't involve immunizing a sheep. And by the way, this isn't even the whole story. This virus that came out had about 1/3 the yield of a typical H1N1 strain. It wasn't until August, it was out here, that we actually got a strain with reasonable yield. So the system clearly was not working optimally. We had at that point started our flu cell culture facility in Marburg. Our Holly Springs one wasn't up and running yet. And one thing really did work. Although this is a bit of an exaggeration because there are more mechanized and ways of inoculating an egg that leaves a somewhat smaller hole. Nevertheless, egg-based manufacturing is fundamentally not a high biocontainment process. It's messy. Flu cell culture can be done in something that looks a whole lot like what you expect a pharmaceutical manufacturing facility to look like. A lot of stainless steel. We're not wedded to stainless steel, but clean and sterile and not with fluid sloshing all over the place is kind of nice. And it turned out to be critical in that year because early in the days of any outbreak, you don't actually know how pathogenic the virus is going to be. And with H1N1, there was a concern because the early reports that came out of South America was that this thing was killing people all over the place. It turned out to not be that pathogenic, although it was for certain groups. But it was treated like a highly pathogenic agent early on out of an understandable precaution. But that meant that our egg-based manufacturing facilities simply sat idle because you couldn't handle the vaccine, the virus, under low biocontainment at that time until people understood its pathogenic potential better. You could handle it here. So as a result, the first vaccines to be tested clinically in Western countries were produced out of the cell culture process because of that containment advantage. We've since now built this giant facility down in Holly Springs together with the U.S. government that has this goal. I don't think I have the number here. I believe it's supposed to make 150 million doses of pandemic vaccine within the first 6 months of the outbreak of a pandemic, assuming that vaccine can be adjuvanted so you can lower the dose enough that you can get those kinds of numbers. And this thing has these giant bioreactors, 5,000 liter bioreactors to grow flu viruses. It's actually a very cool thing to be able to take what is essentially initially a bench scale process and scale it up like this. So you would think you're making your vaccines in mammalian cells. Haven't you gotten rid of this problem of antigenic match? And the answer is no, we have not. And the reason is that egg adaptation is like original sin. Once you have it, you have it. So when you take a virus and adapt it to eggs and subsequently pass it in MDCK cells, those egg adaptations don't revert. The MDCK cells have both alpha-2,3 and alpha-2,6, the human and the avian receptors on their surface. So once you have that egg adaptation, it perpetuates. So although I wish we just licensed our flu cell culture vaccine, and I wish I could say, now the world has a better matched vaccine, I can't say that because it's not true. In fact, we're still using the seeds from the World Health Organization that are adapted to eggs, and therefore the vaccine still has the mismatch. So what do we do? Well, this recapitulates a little bit of what I've discussed right now. By getting rid of the eggs in manufacturing, you get rid of the egg supply problem because you don't need that many eggs to just make the seed. You can produce before a biosafety level has dropped. You can eliminate what we call bioburden, also known as poop, that introduced into manufacturing from chickens. And you can, but you still have the strain mismatch. And you still have all those issues with adaptation, low strain isolation rates, etc. So the next step is to try to move this to actually a fully cell culture process, or as I'll discuss shortly, a synthetic process. I think because I'm going over a little bit, I'm going to skip some of the epidemiology, which is a little bit, although I'll include a little bit of it. This is the epidemic curve in 2009 where you had your first wave, your second wave, and if you look overall, yes, it was more. It was not dramatically more, however. If you looked at the pediatric mortality rate, though, then you saw dramatically more. And this gets into one of the other challenges of flu vaccines, and that is when you try to compare year-to-year efficacy or immunogenicity, you have to take into account not only the changes in the vaccine and in the circulating strains, but the changes in the immunologic history and the infection and immunization history of your subjects. And it matters. Typically, the deaths are in the elderly. for the 2009 pandemic, it was in the very young. And Dr. Monto alluded to this before. If you look back, it was actually a rather extraordinary finding from our colleague James Crow down in Nashville, was that although you need to change strains every few years, because a couple of years of drift is sufficient to knock out the antigenic cross-reactivity. Sera raised against the 2009 pandemic strain cross-neutralized and cross-HAI'd with the 1918 pandemic strain, so across a distance of approximately 90 years. And as it turns out, that's because part of the explanation is that part of the explanation for the relatively low mortality among the elderly was that they had pre-existing immunity from the original 1918 strain and its recent successors. And that immunity actually lasted all the way out here. On the other hand, we got very lucky during that pandemic that we only needed one strain, and in fact you didn't need the adjuvant to get an effective vaccine. For H5 vaccines, for H7 vaccines, you need it. You didn't here. And the reason appears to be that although antibody reactivity, cross-reactivity, was present in the elderly who've been exposed to those very similar strains, T-cell cross-reactivity was present even in young people who had seen other H1N1 strains. And therefore, although the elderly had pre-existing humoral immunity against 2009 were protected, the 2009 pandemic strain were protected, the young had cross-reactive T-cell priming. And so one boost of that priming was good enough for them. With the H5 strains, the H7 strains, nobody's even primed. And so you need both a couple of doses and an adjuvant to be able to prime sufficiently. So there's this wide biological range in the interaction between humans and vaccines and the viruses, depending on the epidemiology and the different arms of immunity that are engaged by infection and by immunization. Getting back briefly to why there's cross-reactivity between a 1918 virus and a 2009 virus, it gets back to the fact that we eat pigs. And the viruses cross over from avian species into humans and into pigs. We don't eat humans. They live a long time, and they get repeatedly infected or immunized, building up immunologic pressure on the viruses that then become endemic in humans like the H1N1 strain. That causes the virus to drift. Pigs are also infected at around the same time. However, they generally don't live to see anything beyond their first or second year because we eat them first. And as a result, there's very little immunologic pressure on those viruses, and they remain constant. And you now have a reservoir of viruses that are little changed antigenically, so that they can come back and reinfect the human herd once we've lost our immunity to those old strains. And so there's this notion that there is a cyclic pattern to pandemics, potentially. H1N1, we've seen it. There's potential for H3N2 to do something similar. And this leads to the notion of pre-pandemic anticipatory immunization. There is some basis to think it might be possible. By the way, I should do a time check at some point, because I can go on forever. I have 5 minutes left. Okay. So I'm just going to talk about these briefly. I'll talk about the synthetic system, and then I'll wrap up. If you don't prime ahead of time, if you just give people a couple of doses of a pre-pandemic vaccine, you get very little. Without adjuvant, almost nothing. 1 to 40 is considered, for reasons that can be debated, protective. If you adjuvant, you do a bit better. However, this is work that we did back in 1993, I believe. And it was with H5N3, not H5N1. We then came back to those people 6 years later and gave them an H5N1 vaccine, a heterologous boost. And if you gave them an adjuvanted boost within a week of getting one boost, you got very, very high, in this case, HI titers. And those titers cross-reacted against all the H5 strains that we had available at the time. So although we don't have one vaccine that can give broadly lasting immunity, we do have systems where if you have a prime with an adjuvanted vaccine, a long interval, and a heterologous boost, you do get quite broad immunization. This is not universal, but it is a lot more strong and a lot more broad than conventional immunization. And this leads to the possibility of potential pre-pandemic immunization where you prime the population with one strain that is moderately similar to what may come out later, and then you are primed so that a lower dose, a more rapid response, a higher response, and a more broad response to a more strain-specific vaccine would come up later. So integrating this to some degree, so now can you now make that strain-specific immunization that could follow less specific priming faster? In the aftermath of 2009, we collaborated with BARDA sponsorship with Craig Venter and his groups, particularly Synthetic Genomics Vaccines, Inc., to see could we now synthesize our own vaccine viruses and do it much more rapidly. The idea is you get the sequence off the web, you can screen the web by algorithms to ask the question, with the new sequences come out, how likely are they to be new antigenic variants, synthesize oligonucleotides, stitch them together, make DNA cassettes, transfect cells, and out come viruses. When we first started doing it, it didn't work. And the reason is oligonucleotide synthesis is just too inaccurate. We added enzymatic error correction of mismatches when you completely synthesize both strands, and we got our error rates down so that in the rescued viruses, more than 90% of them have exactly the intended sequence in their HA and NA genes. At that point, once you have your accuracy to that level, you no longer need to sequence in the middle of your virus synthesis. You can start your virus synthesis, you can go all the way through it fast, and then you can look at the viruses you've got at the end, and 90% of them are going to be right, so you can do your sequencing retrospectively. And the speeds you can get are actually prodigious. From the download of an HA and NA sequence to having fully sequenced genes, synthesized genes rather, is less than 1 day. I won't go through this, for reasons of time I won't go through this, but once you take control of this process and say we're going to synthesize the virus, you can choose what virus you're going to synthesize. So why go along with that boring old PR8 backbone? Why don't you make something that's better adapted and grows better? So we went ahead and we made better backbones that produce more virus. This is what, at the time when we ran into the story that I showed you later, that when we started making our own viruses, we realized the viruses we were getting from WHO weren't very good matches because of egg adaptation. Because we didn't have egg adaptations. We were matching circulating strains better. We were no longer matching the recommended strains so well. I'm going to talk about the system and then show that the system works. The goal is to move from the current system where you're shipping respiratory secretions and viruses between WHO collaborating centers using old technologies, centrally controlling everything, to a more modern system where, particularly in the future as we get toward closer and closer to point-of-care sequencing as a diagnostic tool, right now it's just a research tool, but we're not that far from the point where it's a diagnostic tool. Sequences are posted, are analyzed, viruses get synthesized that are optimized, and in cell culture, well-matched strains get made, and potentially even the new strains distributed between manufacturing centers electronically. This sounds like a little bit of a fantasy, but I'm going to show briefly that it is not. We have done this, we have used this system, and it works. So, on Easter Sunday, 2013, I got an email from Peter Bogner. He's the guy who runs GISID, which is this global system, global initiative for sharing all influenza data. It's a server and a bunch of legal arrangements around it to try and overcome the very interesting and substantial problems of getting people to share their data. That was on Easter. The next day, our colleagues at the Veterans Institute started their gene synthesis in California. By Tuesday, the genes were synthesized and being FedExed to our Novartis Cambridge virology facility. By Wednesday, we transfected and started rescuing the virus. We also shipped the genes to CDC and said, you can send these to anyone you want for public health purposes, which they did, because the virus was only in China at this point. It wasn't being distributed anywhere. By Saturday, we had rescued a seed virus on a series of different genetic backbones, and by Monday we had confirmed that we had the right sequence. It wasn't until somewhere out here that the Chinese started even sending out the virus to start to make vaccine viruses, and it was many weeks after that before there was any success in making any viruses using that. So for a period of time, these genes that we sent to CDC and some others that were made at St. Jude Hospital were the only genes available to make a vaccine. Now, if I stop the story here, it would all look very glorious because we got this very quickly. And in fact, we did make vaccine, test it in humans, and supply it to the U.S. Pre-pandemic stockpile before the second wave of the outbreak. But the rest of the story isn't quite so pretty. This is what I showed you here. Here's where we inoculated our first 2,500 liter fermenter with the virus. What were we doing all this time? A couple things. First, this was still being done in a research lab. Today we now have this all implemented in that Holly Springs facility, all under high manufacturing quality standards. But at that time, we'd done it in a research lab. We had to take that virus and really do a lot of work to demonstrate that this virus is safe to use in making something for humans. But a lot of what we were doing was waiting for permits. So the U.S. government had funded us to develop this system. They had built us a laboratory, basically, to do this kind of work. They'd also built this factory in Holly Springs. And the goal was to make vaccine to protect the U.S. population. But the U.S. Department of Agriculture would not allow us to ship the virus from our Cambridge research facility to our Holly Springs manufacturing facility. So what did we do? No one said we couldn't ship it out of the country. So we shipped it to Germany. And after a little hassle in the Frankfurt airport, it went to the University of Marburg in their BSL-4 lab. And there they did lots and lots of testing, showed that it was safe, and they shipped it to our German manufacturing facility. And there we made the vaccine batch, and then we shipped it back to the United States to do the clinical trial, and we managed to get the stuff for the U.S. stockpile. Obviously, there are people who don't know what the other people are doing, and I think a core message here, and this is for a not highly pathogenic strain. H7N9 is pretty nasty, admittedly, but it's not at the level of pathogenicity of H5N1. All this stuff in the middle is all stuff that we could get rid of. And we've done that in part by implementing everything in one facility. So the only things that need to go into that Holly Springs facility right now is an email with the sequence and raw materials, and non-infectious raw materials. And what we can put out of that facility is a vaccine. Now, addressing the reagents to release that vaccine is another issue, and that's for another talk. But that is the goal, to try and eliminate all this stuff in between. And I think I'm going to skip the rest. But the next generation that we're aiming for is actually having the genetic sequence, in this case a self-replicating RNA, be the vaccine. And I will show you this. So we do have this RNA-based vaccine where the RNA encodes an RNA-dependent RNA polymerase. For the virologists of you here, you inject it, it replicates, it can't spread, it doesn't encode any cell entry or egress apparatus. It actually works quite well. Manufacturing footprint incredibly small. And in 2013, when we were synthesizing those genes that we sent to the CDC, we did 2 additional enzymatic reactions: an RNA transcription reaction and a capping reaction. We mixed it with our lipid delivery system, injected it into mice, and we actually had a vaccine ready to go for mice at this point, 8 days after the sequences were first posted on the internet by the China CDC. So that's where we want to go with this ultimately, is really to radically transform this system to be much better. However, I would say I'm calling these radical, but these would all, I believe, fall into the category of what Dr. Monto describes as incremental improvements. There's a whole other story about our universal flu vaccines possible, and I share the belief that it's not very close. It's not that I don't love the science, that looking at broadly neutralizing stem apoptosis, cross-reactive T-cell immunity, etc. All those things exist. They're all real. They all induce some immunity. They can all be tapped for vaccine. However, our current vaccines tap into the most potent immunity. That is that immunity against the variable head. And we're looking at on the order of 50% efficacy. If you trade any of that efficacy for breadth, what you get is a broadly ineffective vaccine. That doesn't, so I suspect that the initial changes are going to be additions to our current vaccines to give them some breadth, because you don't want to give up the most potent immunity you have. And I certainly don't want to be a nihilist. Long run, for a flu vaccine to be really broadly and potently protective, it would have to give much stronger immunity than a whopping case of the flu. There's no law of nature that says that's not possible. On the other hand, that's a pretty big challenge. So I'll end there before the hook comes out. Yeah. Yeah.